Hi, good morning, and welcome to the ZP Developer Zone. I realize at the moment my camera's not working, but I'll try and fix that during the webinar. But yeah, welcome to the ZP Developer Zone. So every week at 8 a.m. London time, we like to do a webinar specifically for the members of our ZP Developer Zone, and I'll essentially jump straight into it. So if I jump into it for this week, I just want to highlight that we have the ZP Academy. The ZP Academy is a free training course where we put out electrochemical data and information regarding ZP. And I also want to highlight that we have our webinars. We do those every Thursday at 8 a.m. London time, where we answer technical questions. We have our, let's say, job posts. We have our ZP Developer Zone, and we have our workshops. I know I commented on it, but we do have a workshop with Elsevier at one of their conferences pretty soon, and ZP will be providing essentially a webinar, a training course at that particular conference. Now, if I just go on with the slides a bit. So every week we like to answer technical questions. This week, the technical questions that we've had are really regarding one of our an inquiry that's come in through the website, and they're really asking. It's kind of come in bits and bobs, and I've sort of had to knit it together. And what I've knitted together is they want to make a 4-electrode arrangement. So it made me think immediately this sounded a little bit like an impedance measurement. They said that they wanted to measure on 30 to 50 microliter droplets, at which point I started thinking about droplet microfluidics, but we'll return to that. They wanted to measure the dielectric properties of cell suspensions. And with that, we were really when I read that, I kind of thought, okay, this sounds a bit like impedance spectroscopy. And again, I had a sort of another bit of a note that said that they were interested in microfluidic attachments. So I've sort of been weaving this together, my understanding as we've gone along. So dielectric spectroscopy, I'm hoping that they mean, is really mean in the context of impedance spectroscopy. So dielectric spectroscopy for me is a subcategory of impedance spectroscopy. We talk about impedance spectroscopy a lot in these webinars. We talk about impedance spectroscopy in the context of really turning electrodes into biosensors, where we have obviously antibodies anchored to the surface, and then we have antigens that bind to those antibodies, and that's the source of our signal. So we do talk about that a lot in these webinars. So today we're not talking about detecting proteins and antigens. We're here talking about measuring the dielectric properties of cell suspensions. And so I've interpreted that as there's some cells, essentially, I don't know if it's bacteria, mammalian, but there's some cells, they're in a solution. They're obviously, because of their buoyancy, they're essentially in suspension, and the person wants to measure the capacitance and resistive properties of that suspension. They call it the dielectric properties, and I would call it, that this is really a form of dielectric spectroscopy. A dielectric spectroscopy for me is another word for, or another term for impedance spectroscopy. So there is some literature for this, around this, and I found this paper on ResearchGate, and this is from that paper. So I want to make sure that you understand that this is not my diagram. It came from the ResearchGate paper, but I think they do a nice illustration of what's really being asked for here, which is we have essentially an electrode and an electrode, and between that electrode we have a suspension of cells. I think cells are really sort of interesting in this format because we like in electrochemistry use equivalent circuits, And so they've kind of illustrated that across a cell, you essentially have a capacitor due to the membrane. So that's that dielectric property that the inquirer was asking about. You have a resistance through the cell, and then you do have a capacitance essentially on the other side of this cell when the flow of ions has to kind of go through this cell. So we have each cell has essentially its own equivalent circuit. Now, when I read the paper, this was the setup that they had, and I just want to say hi to Hitcham this morning. But this is the setup that they had, where PC, analyzer. They had a cuvette, and within the cuvette they had a couple of electrodes. Now they're using a 2-electrode setup. So they describe it as a 4-terminal Kelvin clip, and they have these electrodes set up. So for me, this is really a form of impedance spectrometer, and I'll touch on this from my understanding of impedance spectroscopy. And as I say, they have this cuvette in which they have the cell suspension, and then they're measuring the electrical properties of the cells by putting electrical excitation between these two electrodes. Now, the results that they had are interesting. There's a few things to comment on the results. The impedances that they measure are actually quite low, which tells me that these are probably uncovered electrodes or unmodified electrodes. It tells me that those electrodes are in the solution because the impedances are so low. It's also interesting that they actually go up to quite high frequencies. So here they go up to something like 100 megahertz. And then they construct a sort of Nyquist type plot of imaginary versus reference. I suppose it's worth saying that ZP does have a lot of webinars and content out there about impedance spectroscopy. So, when I read the paper, they have some Bode plots and they have a Nyquist plot. From the Bode plots, we can sort of see the frequencies that they use, and we can see the absolute or the phase angles, so we get a sense that, their capacitive elements and their also their resistive elements. So let me now, I had an instinct that, in fact, if you have a suspension of cells and those cells have high cell wall integrity, then I would imagine that you'd have a higher capacitance nature. Your dielectric properties would be higher. And I think their data really reveals that when they have living cells, the Nyquist plot shows this kind of larger semicircle. And I think they're basically reflecting the cell integrity. And when the same cells are dead, the Nyquist plot has a sort of semicircle that's a lot less. So I find their results would be in line with my expectation that when you have a high integrity of cell wall, you do have a higher dielectric or higher capacitance. It's because of the cell walls. When the cells are dead and maybe those cell walls don't have the integrity, then the capacitive nature of it decreases. So that was in line with my expectations. The thing that's worth noting is most of the difference between whether you're alive or dead is up here, and so that tells me that they're actually getting most of the useful data in the low frequency range. So I feel like in the paper they actually went up to like 100 megahertz. I think it's ridiculous to go up to 100 megahertz. I think they actually got most of the useful data at the lower frequencies. Now my recommendation would be we're not coming at ZP. We're not coming from an electronics perspective. We're coming from an electrochemical perspective. So the instruments that we recommend are electrochemical in nature. So if you want to measure a suspension of cells and you want to measure their dielectric properties, then in my world that's going to translate over to an impedance spectrometer. The recommendation obviously is the Anapot EIS. Now the Anapot EIS has a LEMO connector here, a 5-pin LEMO connector. I mention it because I would run it, as I've shown here, when actually you plug in a adapter, and the adapter comes to a screen printed electrode, and I'll touch upon that next. So you need the instrument, you need an adapter so that you can then hold screen printed electrodes. We have an adapter, and the adapter comes in this configuration with these thumb screws. If you remove these thumb screws, it looks like the previous picture that you just have an exposed, essentially, electrode. If you put the adapter on, then you essentially have a flow cell. I'll talk about the flow cell in a minute. And now into that flow cell, you can put, we call this carbon 303. This is our gold 303. You can't necessarily put in our hypervalue carbon. The reason that is, I would like you to use our hypervalue carbon electrodes because they're only 99 cents each, but I'm not entirely certain because the materials here are made from a polymer. They're about, I'm guessing about 250 microns thick, whereas these materials are ceramic and they're about 650 microns thick. So I'm not entirely certain that this flow cell will seal on our hypervalue electrodes. So my primary suggestion is going to be the carbon 303s and the gold 303s. The reason I've mixed up the carbon and the gold is because it's worth trying both to see which gives you the best quality of signal. So I'm going to sort of make a recommendation that you try both. Now, the hypervalue electrodes are actually so low cost, you might as well get a pack of them and just try them out, to be honest with you. If it seals well, great, and you get a much lower cost per test because of the cost. Now, the first experiment that I actually try out is to not put the flow adapter on there. I would literally just put a drop on there, and that drop could be something like 30 to 50 microliters, and literally try using the Anapot EIS, try doing an analysis essentially, see what you get. The reason I'm suggesting this is because it's much simpler to be doing this experiment than to do an experiment where you're trying to do it in a microfluidic chamber. You know, it's kind of adding a new degree of complexity. So I would actually just start off simple. Take a drop of the cells, measure the impedance spectrum, let's say, of those cells. If you can induce a death of those cells in situ, even greater, but otherwise, you know, try the same experiment with essentially the same population of cells, but now dead. The second experiment then I would say is put back on that flow cell, because now you're back into what your original question was, which is, I want to measure the dielectric properties of a suspension of cells. Okay, you can do that. You also have some comments that you want to do it in a microfluidic chamber. Well, this creates that microfluidic chamber. And you actually have a flow chamber as well. And this, if you have an artifact in your data, because if you have a static solution and the cells are changing in that static solution, then that might be reflected in your impedance spectroscopy. If you have a pumped suspension of cells, then you might actually end up with a steady state. And so your impedance spectroscopy will then reflect the steady state. But that's I would say almost like a decision that you kind of have to get to. We do, if I was to do this type of work myself, I'd use a syringe pump. This is a syringe pump. I'll link to it in a bit. This one has a valve on it, so you can essentially change the inputs. But if you're going to do a flow microfluidic system, then you probably need a syringe pump. So if I was to summarize the hardware, I understand the question, which is you want to measure the dielectric properties of a cell suspension. Cells have membranes. Membranes, whether the cell is viable or non-viable, will have different, let's say, capacitances, and you should be able to use it in a machine like this to do it. You will need an adapter like this, so that in the first instance you can just put a screen printed electrode in there, and I would drop on that let's say a 50 or 30 to 50 microliter drop of cells and just measure it without any flow. The electrodes that you need will be the carbon 303, the gold 303, and I would recommend trying the hypervalue electrodes as well. If you're going to do the next series of experiments, you'll need a pump, and then all these links that I do here. If you look at this YouTube video, if you look at this video on YouTube, I just want to say hi to Aftab, who's also joined us online this morning. Hi Aftab, hope you're well. All of the equipment in terms of hardware, adapters and screen printed electrodes, I've linked it all here, and including also a pump as well, so you can also see the pump that we use. I'm not necessarily recommending that pump, but I'm just telling you what we use. So in summary, I've read this paper, which I think is close to what your inquiry is asking, and I'm essentially recommending a experimental setup like this. Now, this morning I deliberately did a shorter webinar. We always go up to the 30-minute line with these, but this week I wanted to just answer one question. I want to answer it in fairly good detail, which hopefully I've achieved. I want to say thank you to Hitcham and Aftab. I'm sorry I didn't notice my camera didn't work this morning, but I'm sure I can fix it later on. And thank you very much. If you have any technical questions on Zimmer and Peacock, we do this webinar every Thursday, 8 AM London time. You don't have to watch it live. We will also do recordings. So thank you to the guys who came online this morning. Thank you to the inquiry.